Yes, hello again to most of you. We already met, and I'm happy to be here tonight. And I'm looking forward to an interesting evening, having so many people here tonight. And so shortly for the one who did not know me, my name is Christine Fast. I'm veterinary pathologist from the Friedrich-Löffler Institute and head of the National Referenzlaboratory of Germany. And I'm in prion research for about 19 years now. And most important for tonight, I'm the project coordinator of the scrapie in Iceland project. And that's the reason I'm here, and that's I would like to introduce tonight. We tried in June, but the connection was not perfect. So Angel was so kind to agree to come again to present his talk today. Angel Ortiz-Pelaez is Senior Scientific Officer in EFSA since 2015, and wherein he's in charge of TSE and animal byproducts. And before he went to EFSA, he was a veterinary epidemiologist on TSE at the APHA in UK. And he was working there in monitoring scrapie control programs in the UK, TSE epidemiological research, BSE epidemiological investigations, and so on. That means he has a huge experience about scrapie eradication programs. And we are very, very lucky he's here tonight and will present his talk. Many thanks to you, Angel. The desk is yours.
Thank you very much, Christine, for the kind presentation. Good evening to everybody. I'm going to share the presentation. And I hope that you can see it. Can you see it?
Yeah.
Okay. Good evening. Since we are not going to do a consecutive translation, I'll try to speak not fast, because I usually speak a little bit fast so that everybody will understand me. First of all, just to thank the organizers, the project of Scrapie in Iceland, and all the Icelandic colleagues who are attending this meeting. I was asked by Carolina to give in this presentation a little bit, and Christine, a little bit of a review of the control measures for classical scrapie, and talk about what has happened in Europe all this year that we have been trying to fight the disease. So what I will do is just to very briefly talk about the tools that the European Union has used to control classical scrapie. I will review the implementation of those measures using the EFSA opinion that we published in 2014, when we reviewed the first 10 years of implementation of the control program in the European Union member states. And then I will also use in one of the opinions of EFSA in 2017 about the scrapie breeding for resistance of scrapie in goats, to talk a little bit of what we need to start breeding for resistance in a population, whether it is sheep or goats, and what was the experience that we had when we were studying the breeding for resistance in goats. So without further ado, I would just state that the two main working horses that the European Union has had to control, I would not say eradication of classical scrapie, but control of classical scrapie in the European Union has been, on one side, the surveillance with the objective of detect infected animals in the populations. And that requires that the surveillance must have sufficient pressure and be effective enough to allow the detection of new outbreaks. So animals within flocks that we didn't have information about of their disease status. And then the control of the disease has been based on two measures. One is the consequence of surveillance, which is the management of the infected flocks, what we call the non-genetic sanitary measures, and then the breeding for resistance. So these have been the tools that we have been using for the last 20 years to control classical scrapie. And the other very important tool that we have had is the legal framework. We had the regulation 999 of 2001, what we call the TSA regulation, which is a very big of a regulation, including all aspects related to the control, eradication, diagnostics, feed ban, SRM, related to scrapie or TSCs in general. And this piece of legislation is not static. It has had so far more than 60 amendments, because the evolution of the knowledge and information that we have about these diseases meant to make changes and amendments in a kind of initially increasing the regulatory framework. And then as time went by and the situation of the diseases improved, has been a little bit of relaxation of the measures. So we have a dynamic tool, a legal tool to implement these measures. And what have been the drivers that have taken forward all this dynamic legal framework? Well, basically scientific new evidence. Of course, starting with the detection of BSC, or the discovery of BSC, but then we had issues that made, for example, to start doing surveillance on a scrapie that was not before, when we started to question whether BSC could be present in the sheep population, BSC could be masqueraded by the scrapie epidemic or outbreaks. Then what was the new situation with the description for the first time of a typical scraping on 98, what was the significance of that? And then in goats, then we found BSC cases in field cases in goats. So all these things were a little bit driving changes in the legislation. So what has been the non-genetic sanitary measures? What we talk about the control measures. The situation is that when in the EU, a new case of, and I'm going to say classical scrapie, I'm not going to mention if BSC cannot be excluded or atypical scrapie. I'm going to focus on classical scrapie because I think what matters to the Icelandic colleagues. When a case is confirmed in a holding, there are three options to control the disease, to manage the flock. And the three options has not been implemented simultaneously over at the same time in Europe. Has been a progressive evolution of the options. The first option came with the first enforcement of the 999 regulation, which was the only one that we had at the beginning. Every time we had a case in an infected flock, we had to kill the animals, stamp out, eliminate all the animals in the flock. But we had a situation where we could replace the kill by a slaughter for human consumption. So instead of throwing the bodies to an incinerator or any other disposal, we could slaughter this animal for a human consumption, but with certain conditions. And one of the conditions also is that our animals that were killed or slaughtered, they had to be tested, all animals older than 18 months. And the animals that were too young at the moment of confirmation in the farm, they could be delayed the slaughter until they reach a certain age, which was the usual slaughter time. When the animals were younger than 3 months, they could stay in the flock for a little bit longer until they were slaughtered for human consumption. So this was the only tool that we had at the beginning. Of course, this required, and I remember when we set up this project in the UK, and at that time we had a lot of cases of classical scrapie, was a huge operation put in place from the logistical point of view, administrative, financial, data flow, to implement these programs, to allow the farmer to take the entire flock to a specific abattoir or to a specific knacker jar or rendering plant where the animals had to be humanely killed, and then take the samples from the animals and then take the samples to the lab, etc., and then pay compensation to the farmers for each of the animals. This was reviewed, and in 2003 we had the option 2, which was the substitution of killing all the animals in the flock or taken to a slaughter by only the animals that are genetically susceptible to a classical scrapie. So basically, the breeding rams homozygous ARR and the ARR breeding ewes not having the VRQ allele, etc., they could remain in the flock. And from 2019, the same was applied to goats. So animals having the polymorphisms in the position of the prion protein 222 or 1FOX6, they could stay in the herd. Again, the slaughter of the animals, of the susceptible animals, could be replaced by the slaughter for human consumption within the territory with further testing. They could delay, again, the slaughter lambs for a few months. And very important, a third clause in the regulation, which is that the countries could request the Commission to delay the kill of the slaughter of up to 3 years, arguing that there were very low numbers of resistant animals in the population, and also for economic reasons. And I know for a fact that one country, for example, applied for this derogation. It was the case of Cyprus, because it had a huge problem of classical scrapie in the goat population, and the regulation said that they had to kill all the infected herds, which meant to cull about a third of the population at that time. And for economic reasons, the country argued in the Commission that they were needed this delay of 3 years, and they were consented, but it was also requested to pursue the investigation and the research on the breeding for resistance as an alternative to cull all this large part of the population. And again, there are other requirements, like the non-ARR rams. If they stay in the flock, they have to be killed or castrated. You couldn't have breeding ewes that do not have at least the ARR allele and not the VRQ, etc. So this is the option 2. And then the option 3 appeared much later in 2007, when even the genotype and cull, the option 2, could be replaced by not killing any animal. And in order to get approval or the competent authority to implement this strategy in the flocks, they had to argue some reasons. For example, that it was very difficult to find resistant males in the population so that they could start doing breeding for resistance. I'm talking about sheep. Exactly the same for the goats. Or, for example, they could argue that if they kill and destroy the animals, they will have a problem of inbreeding because they wouldn't have enough genetic diversity in purchasing live animals to restock. Or due to epidemiological factors that could be due to, for example, small breeds in isolated areas with very strong geographical barriers, with no contact with other sheep populations. So this is the option 3, which is mostly what is applied nowadays. There are no killing of animals in infected flocks, but the option 3 is applied. And I want to say this because it's very important that if the restrictions applied to flocks under the option 3 are not complied with, the competent authority can revoke the decision and apply one of the other 2 options at any time. And I have experienced this case because once you have applied this control measure, and you can do it in one or more flocks under the same holding, and you can decide due to epidemiological investigation whether, for example, if there are sheep a flock and a goat herd in the same premises, which one you want to put under the control measures. You can do it more, and you can do it from, for example, apply the measures not to the flock where the case was found, but, for example, where the case was born and raised, because maybe the animal had been in the holding where the case was confirmed very short period of time. So we argue that the animal probably would have been infected in the previous holding. So you can also apply control measures to holdings that are not the ones where you found the cases. And then during 2 years, you have to apply intensified monitoring. All the animals slaughtered for human consumption and the animals found dead on farm, they have to be tested for scrapie. They have restrictions for restocking animals, and also restriction of movements. And this, for example, the experience that there were farmers that were under restrictions and we didn't kill the animals, but they were not respecting the movement of animals. For example, they were taking their flock to common grazing pastures where other flocks, they were sharing the land with them. And that was a very high risk practice. So we had, for example, I remember in the UK, we had to revoke the implementation of this measure and then apply the other ones because the farmer was having high risk practices and it was putting at risk other flocks in the neighborhoods. So these are the three options. And then, as I said, you need to apply all these extra conditions during a period of 2 years. So these are the three options for the control of the infected flocks. Now, when we go to genetic breeding for resistance at the beginning, when scientifically it was really evident that we have an allele, the ARR in the sheep populations of many breeds present in the continental Europe that conferred sufficient level of resistance. Now the problem was how we do that, how we use that as a tool to control scrapie. At the beginning, the commission was hesitant. So the only thing they did was to commission surveys in the member states to estimate the frequency of the ARR allele in these populations, in different breeds, and whether it was present in homozygous or heterozygous, if they were present in rams or in the female population. So to get a flavor if the implementation of breeding for resistance was feasible. And then in 2004, the Commission, in an amendment of 999, obliged the countries to have in place, breeding for resistant programs in what we call the high genetic merit flocks, basically the pure breed sheep, the top layer in the structure of the sheep population. And the regulation defines how these breeding for resistant programs should operate. They need to have a very clear certification system. Each animal has to be individually identified, a centralized database. There have to be clear rules for sampling, for genotyping, the rules for participation of the flocks that to be very clearly stated. And they have to be provisions for the protection of the breeds, if they have rare or endangered breeds, and also study and investigate whether the breeding for resistance can affect the production traits. And this was the main concern at the beginning of this story. I remember in the UK, we spent a huge amount of money investigating if, for example, breeding for the ARR allele in the sheep population would have detrimental effects on, for example, survival rates of the lambs, pregnancy rates, number of lambs per ewe, milk production, anything that could have a negative impact. And these studies lasted for a long time because you need to follow flocks for a long time and the animals individually and take a lot of measurements. And the conclusion was that we didn't have any evidence that the ARR allele was detrimental for the production traits, even though some farmers argue that was the case. But the point, going back to the breeding for resistant program, is that the countries were forced to have breeding programs, but the participation of the farmers was voluntary. They didn't need to join the breeding for resistance program if they didn't want. Only 1 year later, the Commission amended the regulation and made compulsory the implementation of the breeding for resistance in the high genetic merit flock to every single flock in the member states. However, and that's the interesting thing, this obligation only lasted 2 years, a little bit longer than 2 years, because in July 2007, the breeding for resistant programs became again voluntary. And I think that this has had a huge impact on the success of the breeding for resistant programs in some countries. Overall, the genetic for resistant programs were aimed at increasing the area allele, reduce and eliminate the VRQ, without taking much care about other alleles, like the ARQ, the wild type, which was a little bit left aside. So this was the other, the story of the genetic breeding for resistance. So now I'm going to give you some of the conclusions that the review of the 10 years of implementation of the control programs in the EU was done by EFSA, and we published this opinion. In the presentation, you have the link. You can access it, and I recommend that if you are interested, you have a lot of information. Of course, I would say that the surveillance data and the analysis of the data was up to 2014, but unfortunately, not much has happened since then. So I think that many of the statements and the conclusions of the opinion stand valid up to date. So when it comes to the not genetic sanitary measures, what were the conclusions? Well, the conclusion was that if you apply control measures, the option 1, 2, and 3 in the infected flocks, these measures are very effective in reducing prevalence. And it was demonstrated in countries like France or Netherlands, even the UK, where they have very strong surveillance. Because the problem with the non-genetic sanitary measures is that if you don't have a strong surveillance, as I mentioned before, you will not be able to have a successful management of infected flocks. So France, for example, Netherlands, they went beyond the EU requirements in terms of surveillance. They used to test many more animals than the quota. And this allowed them to identify a lot of infected flocks in the early years, eliminate them so that the infection pressure in the population was reduced. And that was very successful for some countries. So this only could be done for sheep until 2019, where as a result of the EFSA opinion, the Commission included the polymorphisms in the goat population. So again, the surveillance pressure is essential, and it has to be constant over time, and most importantly, if it doesn't have a sufficient geographical coverage, you risk to clear or to reduce the infected pressure in certain areas, and to have pockets of populations that you don't know anything about, where a scrapie is circulating freely. So that's another important issue. And as I said at the beginning, it's very difficult to enforce and monitor, because from the logistical, legal, administrative, financial point of view, it's very demanding. And that's what happened in some countries, that they didn't implement these measures strongly enough, so that we are still seeing that there are the countries with problems with classical scrapie. It's the case for, and I will come back to this point a little bit later, the countries with problems with classical scrapie still today. The other downside of this measure is that the agent persists in the environment. Even if we apply cleaning and disinfection measures, all the competent authorities have protocol for cleaning and disinfection, for restocking, the movement restriction, still the agent persists. And I think that Iceland has a very long experience of this issue, so I don't need to stay there. And another conclusion was that if you apply only these measures, it's very hard that you will end up, let's say, eliminate, and we wouldn't say eradicate, but eliminate the cases of scraping the population. We included 2 case studies in the opinion, one from Iceland and the other one for the USA, as examples of countries where only the non-genetic sanitary measures were implemented. And we got full information from the countries, and it's an annexes in the document. I don't need to go much into detail because you are much more familiar with that than myself. So these are basically the main, let's say, headlines of the evaluation of the non-genetic sanitary measures. When it comes to the genetic breeding for resistance, we evaluated 16 programs from 17 programs from 17 member states. And we acknowledge that the advantage of the breeding for resistance is that you build in a similarity to a vaccinated population. You build a level of resistance in your population by increasing the frequency of your resistant alleles. And then the beauty is that if these animals are exposed to the agent, they may not be infected. And if they are infected, they may not develop the disease. Most likely, they will not have involvement in peripheral tissues, which will determine the horizontal transmission. So it will reduce the probability that the animals will infect others. That's the advantage of building a population with high frequency of resistant animals. As I mentioned before, if you don't implement breeding for resistant program very well designed and long-term implemented, then you will end up having poor results. And we have examples of countries like Romania or Greece where they had breeding for resistant programs in the early years, but they have not been constant and strong because of a lot of reasons: financial, lack of resources in the veterinary services, etc. And on the other extreme, if you apply breeding for resistant programs, not only in the high genetic meric flock, but in the entire population, you will be much more likely to increase the frequency of the level of resistance in the entire population and reduce the incidence of classical scrapie. And we have the example of the Netherlands and Cyprus. Netherlands decided to go for breeding for resistance in all breeding flocks, not only on the pedigree flocks or the high genetic meric flocks. And the same happened with Cyprus. Cyprus had a problem with the scrapie in the sheep population and the goats together because there were a lot of mixed flocks and herds. And they started with just a single homozygous ARR ram in the population to start using it for breeding and cascade it down to the entire population. This can only be implemented, of course, in countries with very homogeneous sheep populations, where you have maybe one single breed, when you have geographically homogeneous, and then it's much easier to implement the results. And what happens, for example, with countries that don't have this national coverage, that the situation doesn't improve? It's the case of Italy. Italy has very good breeding programs in certain regions, in Sardinia, in Sicily, for example, but not in the continental Italy. So they have reduced the incidence of classical scrapie in those populations, but not in the rest of the country. And then there is, of course, it takes longer and more difficult if you have a low number of resistant animals to start with, and you don't use them properly. And this is the key issue, that you need to take control of how you breed the animals. Even if you don't have a luxury of having a lot of animals, for example, in sheep with the ARR allele in rams, then you need to control very well how you use it for breeding. And the other key parameter of the breeding for resistant program is to have very effective dissemination. You need to have very clear rules whereby the animals that you are building resistant in the high genetic meric flocks, in the breeding flocks, are cascaded down to intermediate layers, other breeding flocks, and then until they go to the commercial flocks, depending on the layers of your population. So that at the end, you force the use of this ARR allele in the different levels of the population progressively, and you eliminate animals that you don't want to be used for breeding at all. And that was a very important issue as well. At the beginning, I remember that when we tried to eliminate all the rams having the DRQ allele, there was a big problem. No, now we are going to do how we are going to breed. There was just, you know, this reaction by the farmers. But if you provide advice, if you provide support, this can be easily overcome. So that's some basically the main conclusions of the review about the breeding for resistance in the Europe. I just wanted to give you a flavor of what is the situation in Europe at the moment, in the European Union, sorry. We have gone from having almost 2,000 cases of classical scraping goats and over 1,000 in 2013 to be now in the range of 200 cases in goats and about 500 cases of classical scraping sheep. But the most important thing is that only about 20% of these cases that we confirm every year are from new infected flocks. So they are not from the testing of infected flocks. And that's the index. This is the index that we use to see how is the evolution of classical scraping in Europe. If we reduce the number of new outbreaks, new infected flocks, then that could be a good sign that we are reducing the disease in the population. But of course, it has to be in parallel with the surveillance. And the fact is that we are reducing the number of animals tested every year. The trend is statistically significant. We have gone down in sheep over the years, which is very good news. But as I mentioned before, the classical scrapie problem in Europe, in the European Union at the moment, is basically in 4 countries. The rest of the European Union is mostly clean of classical scrapie in terms of detection. Doesn't mean that there are cases undetected. 99% of the cases that we confirm now are coming from 4 countries: Greece, Spain, Italy, and Romania. And the case load in goats is coming mostly from Cyprus, because Cyprus had this huge problem, but has progressed enormously with the management of the flocks and the breeding for resistance. You see that these numbers that we had in 10 years ago of almost 2,000 cases of classical scrapie cases in a year, were 70% of them detected in Cyprus. And this sharp decline that you can see from 2013 and 16 and onwards is mainly due to the successful control measures applied in Cyprus. And then finally, just want to give you a flavor of what we did with the work when we were asked by the Commission, can we use any of the alleles that are in the scientific literature to design and implement breeding for resistant programs in goats in the European Union? So when we face this problem in EFSA, we've gathered with a lot of experts, and then we decided to develop a kind of tool that will allow us to put the tool, to put the alleles that we have in the literature and apply the tool and see what would be the result of them. So what we did is to create 2 levels, a 2-tier tool. The first one was categorizing the number and the level of studies for which we have results for each allele, starting from the simplest one, which are the in vitro conversion studies, so basically looking at the prime protein holding the polymorphism of interest, and to see if with the new methods, the PMCA, etc., they are able to convert abnormal PAP into the normal PMP into abnormal. And then we go to the other extreme, where we have full range of studies, from epidemiological studies, looking at experimental challenges in the natural host, so in sheep or in goats in this case, using multiple isolates to give us the possibility of studying if the allele confer resistance to multiple strains, intracerebrally and orally, if we have bioassays in transgenic mice, and also, as I said, the in vitro conversion studies. And then in between, you have combination of the different studies with different levels of complexity. And then what we did is to apply kind of traffic light tool, whereby we assess for each of the studies what was the results. And we look at, for example, attack rates, incubation periods, conversion, report from field cases with animals with the alleles of interest, and declare if it was red, meaning that the result didn't give any level of resistance. For example, if you have a lot of cases in the population with this allele, then that's it. If you, for example, have the same or even shorter incubation period or attack rates when you do inoculation studies compared to the wild type, then there's no resistance. We go to the other extreme, which is the green, when we don't have reports of field cases with that genotype, or, for example, very rarely we have found an heterozygous animal. And amber, when you have results in between, you have, for example, poor conversion. For example, you have attack rates or incubation periods longer than the wild type, but still you have animals that go down with the disease, either if this is the sheep or the mice. So we combine the two things and we put all the alleles that we had in the literature, and these were the results. So we saw that for the 146 and the 222, we have different level of studies, but all the results were green. So they were giving us reassurance that the alleles confer resistance, even if we didn't have the full battery of studies for 146. Of course, the wild type was in the extreme, so no resistance whatsoever. We have other alleles with not a lot of studies and not showing any resistance. And then we have other alleles with the same amount of studies than the 146, but with dubious results. They were not under the green category. And then the most important or very important thing is that we compare these results with the knowledge we have for the ARR allele when it was included in the legislation as a breeding for resistant tool. And this was the surprise for us, because in 2001, when the ARR was included in the legislation, there were not many studies done for the ARR allele. We had evidence that we didn't find cases of a classical scrapie with the ARR allele, very, very few, type 2 and SP2. And then in the vitro conversions, we didn't have conversion. But the interesting thing is that in 2017, when we did the study, we had more evidence because studies had been done in that period of time, but we were only at the level of the 146 allele. So I think that this has a very interesting takeaway message, which is that you don't need to have a full battery of studies maybe to start using an allele for breeding for resistance, but you need to be sure that this allele confers resistance. And that's a very important issue sometimes we overlook, because in the scientific literature, you get papers about this allele, the 143, the 142, etc., where they describe certain level of resistance, but can you turn this evidence into a real tool to breed for resistance and to put in motion a program at regional or national level where you want to start breeding and disseminate this allele in the population? That's the key issue. So this was a very interesting exercise, because as a result of that, now we can do breeding for resistance in goats as well, using these two different alleles, which and also it's important because we are monitoring with all the cases of scrapie that are confirmed in Europe. It's compulsory to do genotyping. So now we are starting to get data from the member states with cases of the genotype of the cases to reassure us that we don't have cases in animals with these genotypes, or at least if we have cases, they are heterozygous. They are not homozygous in the KK or SS or DD. So with that, I just stop. Sorry, maybe I have extend a little bit my time allocated. And I just put the links to the 2 documents in the presentation that I shared with Carolina. So but anyway, you can find them by Googling, and you can download them because they are in the public domain. And well, that's pretty much what I have to say. Thank you very much. And I don't know if you have any question or comment, or at the end of the session, I'll be happy to try to answer them. Thank you.
Thank to you, Ange, for the very informative and interesting talk. So if anyone has questions, please raise your hands or just start talking. We are not so much, so you can just start talking as well. Sigurbo and Sigurbjörn?
Yes, thank you. I thought this was very interesting. I saw you presented a relative risk of 0.97. That was statistically significant at 0.01 level, which I thought was quite impressive because it's only 3%. Yeah. But what I didn't quite get is what was done to get this reduction per year? Is it those 3 steps that you were presenting, or did I miss something?
No. You mean the reduction of the cases?
Yes.
Yeah. It's the implementation of the measures by the countries. I mean, all the countries in the EU have to implement the management of the infected flock, as I explained. So by eliminating or the susceptible animals, or killing the flocks, or monitoring the infected flocks, and then also by the effect of the breeding for resistance. So if we see this trend, it's the effect, not only because the graph, it's only for the last 10 years. I should have shown you the graph from 2002 when we actually started. So we should have shown where the cases were in 2002 and go down over the years, which has been really a substantial decrease. So yes, it's the effect of the measures. And we had discussions of what was most important, the management of flocks or the breeding for resistance. And, these are not clear conclusions. Which weighs more? There are people who think that breeding for resistance is much more effective because the management of flocks is much more complicated. But in the other side, in certain countries, to implement breeding for resistance is very complicated. For example, where I worked for many years in the UK, the number of breeds that you have in sheep is, you cannot imagine. With a very multi-layered structure of the sheep population, breeding, not breeding, mixing, creating F1s, F2s. It's complicated. So to do breeding for resistance, trying to include all these breeding layers, it's very difficult.
Thank you.
Jörn?
Yeah, Angela, thank you very much for your very good presentation. Can you, on one slide, you presented that the trade restrictions were part of the control measures. Can you say something about that and the problem it causes in some countries?
Which problems? Sorry, I didn't.
Problems in trade restrictions. If you cannot trade non-resistant sheep.
Well, basically, the problem is that once you, you mean in the infected flocks or in general in the population? In the infected flocks, movement, trading within those infected flocks, or in general in the country?
In general, from infected flocks or to infected flocks.
Well, there is a restriction because, if, for example, now that when you apply the option 3 and you don't kill the animals, you cannot move the animals, sell the animals for life easily. You are under restriction for 2 years, where you cannot freely trade live animals to other flocks. You are under restrictions. The movements are restricted, so you cannot, for example, sell breeding animals for the sake of selling breeding animals. This is something that it's been also subject to a lot of discussion, because at the beginning it was 3 years, then it was reduced to 2 years. And if you consider that you don't kill the animals and you are only monitoring and you are testing only the animals that are found dead on farm and in the abattoir, 2 years maybe is not enough to keep the control measures. So if you free the flock after 2 years and you let them trade freely, maybe you are exporting, let's say, from the flock infected cases that you didn't detect. And this has been a concern that we had, and we have experienced clearing flocks, restocking living animals, restocking, and then appearing again the disease. And you don't know if it was because of the animals that stayed in the flock were infected or came from outside. So it's very complete. It's difficult to ascertain. I don't know if I answer your question.
Yes, it is perfectly. Thank you.
Thank you.
Okay, Asa? Yeah.
Yes.
Yeah.
Thank you for this. You said that in the Netherlands they had a great success in breeding.
Yeah.
And how did they do it? How did they get most of the breeders to take a part, and what was the key of this good success?
I think that the key to good success is that they involve the farmers, and that was the issue. In Netherlands, the sheep population is not large, and they could involve the most of the farmers into the program. Then they have homogeneous sheep population. There are not a huge variety of breeds, and then they give a lot of incentives and support for the breeding and to mobilize the resistant animals, to provide support to breeding, to maximize the breeding using, for example, artificial insemination, etc. And the good thing, maybe that's most important, is that they didn't support only, as I said, the high genetic merit flocks. They involve every single breeding flock in the country. And that's why, because they set targets in the Netherlands, because we had a Dutch guy in the working group, and he was the guy doing the model and estimating what would be the minimum ARR frequency that the population should have in order to expect not to detect more classical scrapie cases. And they were insisting, implementing the program until they reached that level, which was, if I don't remember well, over 70% of the animals holding the resistant allele. So I think that Netherlands is an easier place. Like, for example, where I mentioned Cyprus is another example with the sheep and with the goats. It's monobreed, relatively small population, very uniform, not a very large country, and they will put resources in the veterinary services to implement the breeding for resistant program, and they eventually involve everybody. That's what I could say.
Thank you.
Okay. It's possible to ask in Icelandic if someone else has a question, and ask in Icelandic and you translate for Angel, maybe, if there is the necessity.
Já. Einhver hérna sem vill bera fram spurningu á íslensku?
Everyone is happy.
Everyone is happy. Everyone is
I have still a question. I was talking with Romano lately, and we both realized that we in Germany and Italy have totally different way of processing after classical scrapie case, and I was wondering after that if there is somewhere a list with the different methods the different European countries apply. Do you have something like this, or is it in the internet? Can I find it in the internet?
You mean the control measures that they apply in each country?
Not the controlling, but when I have a positive case in a German flock, in Germany, the whole flock would be genotyped, and all the animals from which are without an ARR allele will be culled. And Romelu told me they didn't do that in Italy. They have another, they process it differently. So I was wondering, we were both surprised about our different regulations. And so I was wondering if there is a list how the different European countries process outbreaks.
It is not a different legislation. Actually, Germany is complying with the regulation, and Italy is complying with the regulation. The issue is that Germany is applying the option 2, and Italy is applying the option 3.
Yeah, I know that, but it's somewhere a list. I can see which country applies which option.
In 2014, we did a survey in all member states, and we asked them which measure you apply, what do you do when you confirm a case, how you manage the flock, you apply option 1, 2, 3, what is the proportion of flocks that you apply the genotype and cull or the other measure, how long you keep them, how do you implement the intensified monitoring. And we collected that information, and it's summarized in the annexes of the opinion. So if you look at the opinion, you will see in the annexes a lot of information about the answers to these member states. Okay. And you will see that the member states implemented within the rules of the European legislation with certain variability, quite variability. And I can tell you, for example, that when option 3 came, many countries, they were straight to option 3 because they didn't want to kill animals. They didn't want to upset farmers. They didn't want to die, and they went for option 3 because they thought, okay, we keep the flocks under restriction 2 years, we test the animals, and that's it. But other countries decided, like Germany, to do it in a different way. But it's all recognized in the regulation. The 3 options, you can apply any option. I think that there were still countries 12 years later that they were still applying option 1, so killing the entire flock, which I think is what Iceland is in a way doing or has been doing. So it's up to the country. Look at the opinion and maybe you will get this information.
Okay. Sigurborg?
Yes, thank you very much. It was very interesting. We are just in our first steps by using the new method, the breeding method. You mentioned Netherlands and Cyprus as a country that have good results. Are those the countries that you recommend Iceland to look closer to and perhaps copy their programs? I can also say I have already called for information from the CVOs in Netherlands and Cyprus.
Okay.
So I've already started, but I would like to hear your opinion.
I think so. I think that there would be a good examples. I was involved in the breeding for resistance program in Cyprus. I follow, and we were advising the Cypriot colleagues to implement the breeding for resistance. We follow all the studies and the implementation, and the results are really interesting. And I have to say one thing, that Cyprus, for example, does a very particular thing that not other countries do, which is that they genotype every single breeding animal. So once the lambing season comes and the farmers left a certain number of animals for breeding, female or males, they automatically register them and they genotype them. And they put them in a national database. So when these animals are moved from one flock to another, they know already the genotype automatically. And if by any means they find any animal with a genotype that they don't want, the animal is confiscated and is eliminated. So, and that is an effort that I think Netherlands did as well, or was very close to do that. And I think it is very important when you have a relatively small population, a uniform, a homogeneous population, because that will give you a full control of how the breeding for resistance is working and the evolution of the dissemination of the resistant alleles in the population. I think it's, and Netherlands did very well, actually, did very good program. And I think that there are two good examples. Definitely.
So,
Isa?
yeah. So for how long time did they do it, genotyping every animal? Because after some time they had homozygous rams, and then they perhaps didn't need to genotype all the offspring.
They genotyped only the breeding animals, isn't that
It's
right?
only the breeding animals. The animals.
You mean only when they started? Then they started with all the breeding animals.
Mm-hmm.
Yes. Yeah. At the beginning, they did a survey and they genotyped all the adult animals, all the breeding animals in the country, because they wanted to identify resistant animals. And this was very important because that will allow us to define a very good strategy of how disseminate the resistant alleles. It's not the same if you have 5 homozygous rams, for example, that you have 20 heterozygous, or you have 2 or 3 or 200. It changes enormously the possibilities and how you should design your rules for dissemination. So they did this blanket genotyping of all the breeding population, and then they detected the animals, and then they bought the animals. They passed a rule whereby they took control of those animals. They took them to a reproduction center that they built, and they started to control completely the breeding with those animals, to maximize the genetic potential of those animals by extracting semen, doing inseminations, controlling that, for example, these animals would only breed with heterozygous ewes, so that you will ensure that you may have some offspring homozygous, and then you would have another homozygous ram, which was the target. The target was to focus on producing resistant or semi-resistant males, because they, of course, they disseminate more than the females in the population. And then they started to use those pool, and then when they had sufficient numbers, they let them stay in the flocks, because they had sufficient numbers. So if a lamb was born, a male lamb was born in a farm, ARR, if they had enough, then they told the farmer, you can keep this ram and you can use it for breeding in your own farm. But of course, to reach this point, you need to have a sufficient number of resistant breeding animals. And for example, if you look at the reports that we publish every year with the TSC surveillance data, we also provide data on the genotyping of the countries do in the random population, in random chip, to monitor the evolution of the genotype in the general population. And you will see that we receive every year from Cyprus between 65 and 70,000 genotypes, because these are the new genotype animals in a year. So they add about between 60 and 70,000 breeding animals to the national herd, fully genotyped. And they have been doing it, you ask me. Well, I can tell you that when we started to go there was 2009, and they were starting to do the full genotyping with money from the government. So yeah, they have been doing it already maybe 13, 14 years.
No. Was it like that in the beginning, that the farmers, they got heterozygous rams, or did they start in the beginning only get homozygous rams from this breeding farm who was producing?
Yeah, they were because they were focused on the 146 because the breeding in Cyprus is a breed that is not present in the rest of Europe. So that's the 146. So it was, I think, at the beginning they found, I don't remember well, but the majority of the animals that they found with level of resistance were heterozygous. They were not homozygous. We published this. We killed infected flocks and we genotyped them entirely. And in the surveys that the country did, they didn't find easily homozygous DD and SS. The majority of the billy goats that they detected at the beginning, they were heterozygous. They were ND or NS. They only had the one allele. So it was not easy at the beginning.
I was still thinking about how was the plan the farmers were working after when they started to use this breeding animals. Then they must have been supported to genotype the offsprings to select the right.
It's free. It's free. The government does the genotype for free, so that ensures that all the breeding animals are genotyped. So the farmers do not pay for that. Is
it still like that? And is that maybe everywhere in Europe?
No. It's only in Cyprus. No. I don't know at this point in time if there is a country, a member of the EU, that is doing systematically large genotyping of large number of animals in the country. I don't think there are, to be honest. I know for sure that countries like the UK, Spain, France, large countries with large sheep population, they are not genotyping animals at all. And actually, one of the things that we did in the UK 10 years after the end of the breeding for resistance was to do a survey in the sheep population to see if maybe we were going backwards, and because there was no breeding, the proportion of animals with susceptible alleles was increasing again, because the breeding for resistance had stopped 10 years earlier. So we did this survey, and the results were not very good, actually, because we addressed the entire population, and there was still a proportion of animals with a strong susceptibility. We have VRQ animals in the population. So I can tell you that at the moment, most of the countries in Europe are not doing a lot of genotyping.
So I shared a link with in the chat where you can find a detailed description of all member states for different years with control programs for TSE. So maybe you can find some details there too.
Yeah.
One last question. When Netherlands and Cyprus are the best examples, what about Italy or Spain? Why they still have problems with scrapie, with classical scrapie? I think in particular Sardinia have a lot of problems. And what's the reason after 15 years of breeding for resistance to having still so much problems?
Well, I think that Italy, as I mentioned before, actually Sardinia has improved the situation a lot because of the breeding for resistance, particularly for the Sardinian breed, which is the one that is in the island. And the same, they have some breeding for resistance program in the Isles, in Sicily and in the south of Italy, but not in the rest of Italy. And that's, again, if you don't implement a really comprehensive national coverage of breeding for resistance or control measures, you will not get rid of scrapie. And this is what happened in Italy. In Spain is more an administrative political issue. There are 17 regional governments with authority or to implement the breeding for resistance in each region. So if you don't have, it's very difficult to have an homogeneous implementation of any control measure when you have devolution of the authority or the power to regional governments, because they don't have the same level of resources, they don't put the same the resources, the priorities. It's exactly the same situation like, for example, in the US. In the US, we don't have a clue what is happening with the scrapie in the USA, because each state is completely independent of the neighboring one. And the policy to control animal diseases in one state could be completely the opposite than in the neighboring state. So this happens with chronic wasting disease and scrapie, and that's the result. So I think that's the problem. And Romania, because it came on board of the European Union much later, and they implemented the measures much later. And Greece has a problem with the resources. Greece has been arguing it's not complying with the regulation, and I am in contact with the Greek colleagues, and they call me and they say, Angel, we cannot send you the data this year because we have an outbreak of lumpy skin disease, we have an outbreak of this disease, and we don't have vets to do the testing and the sampling for TSCs. So it's
so if you have, so would you say if you have a good governmental support of the control, of the eradication and so on, what's the key for successful eradication?
Absolutely. I think the institutional support, the legal framework is very important, because then it's a rule for everybody, it's stiff for everybody. You don't make exceptions, so the rules will apply to everybody, and everybody could benefit of the programs. So I think that the institutional support is important, but also all depends on what is the relationship between the institutions and the farming community, which is a completely different topic, and it also has an influence. What is the relation between the institutions and the farming community? In some countries, they don't trust them. They came always to take taxes. They don't support the farming community. You know, they have this mentality. Yeah. But when you get them on board, if you get on board the farming community and you really demonstrate that you are committed, I think the program should succeed.
Okay. So any more questions? Otherwise, I would go on with the presentations. Yes.
Sorry for the length, maybe.
No, it was really informative, and that's the thing that we wanted to have a talk here. It was really good, and I think I speak for everybody that you brought a lot of information to us. And again, Ange, many thanks for your kindness to join so late in the evening. You are invited to stay, of course, but if you want to enjoy the rest of your evening, we would understand. And yeah, thank you very much.
I will stay. Thank you very much for inviting me, and I will maybe follow with some of the presentations to learn about the project. Thank you.
Yeah, Sigurborg, you have a question for Ange?
Yes, no, it's not a question actually. It's a comment, because I totally agree to Angel. It must be a cooperation between the government and the authorities and the farmers, and otherwise it will not happen to eradicate scrapie. So we have already begun to work on that way, and I have already asked the farmer unions, the sheep farmers, how will they see the plan? I think it's best that they will say and speak out what are their expectations, and how will they do it, and then we can continue on that plan. So just comment on that.
Okay, so I would go on. We have the problem that now I see him. I only give you up, Juan Carlos. Can you hear us and see us now?
Yes.
Yeah, perfect.
Yes.
Okay.
Hello, can you hear me? To me?
I lost contact for about 40 minutes or so. I'm glad you are
Yes.
here, Juan Carlos.
I was here.
We go ahead. I will start with a short overview of our scrapie in Iceland project, and Juan Carlos and Jörn Gethmann will make the biggest part of the talk. So we are a little bit late now. I will start immediately. The scrapie in Iceland project, most of you already know it. It's was funded last year for 3 years, and for us it was important when we applied. It was important to understand the exact measurement mechanism of prion strain and evolution under natural conditions, and for us as well the biochemical and biological factors that may determine zoonotic potential of a prion strain. That was the basic of our application, but this comes with a lot of additional benefits also for Iceland, and that I will explain. Why is Iceland, scrapie in Iceland, so special for us? There's only one breed of sheep in Iceland that was introduced about 1,000 years ago, and which was more or less genetically isolated since then. And we have regions with high natural infectious pressure. When we scrapie was introduced in Iceland about 150 years ago by one single animal. So all the diversity we see today is dating back to one outbreak. And in the past, the cull sheep were often buried or burned close to the stables or in the meadows. So the reoccurrence of scrapie in affected farms indicated that the contaminated places might exist. We are 8 partners from 7 countries. From Germany, it's myself and Jörn Gethmann, as well as Gesine Lüken from the University of Gießen. We have John Spiropoulos from United Kingdom, Fiona Houston from Roslin Institute in Edinburgh, Vincent Bering from INRAE in France, Juan Carlos Espinosa from CISA in Spain, in Madrid, Romulo Nonno from Rome, and Stefania Torskesdottir from Iceland, from Keldur. And the project consists of 4 interactive work packages, so we were working closely together in exchanging results. And as you may remember, in Iceland, we already presented the parts of work package 1. That was the survey of the PRP gene variation as well as the in vitro examination. So it's recorded, I guess. So today we will focus on the in vivo modeling of protective effect of the prion protein gene variants identified in Icelandic sheep. And this will be done by Juan Carlos. So Juan Carlos, the desk is yours. Just a second. So.
Let's see.
Does it work?
I cannot. Yes. Can you see my screen?
Yes.
Okay, thank you very much. Good evening again. In order to study the protective effects of PRNP polymorphic variants, the utilization of transgenic mice offers several advantages when comparing to the use of the chips. We can study easier the individual role of each PRPC variant, because in sheep we have the coexistence of the different variants that we need to breed the animals in order to study the one that we want to work with. We can avoid the effect of other genetic factors of the sheep this polymorphism may be in low frequency, and there is a genetic mixture. We can study a wide variety of prion isolates in transgenic mice because it is less time consuming and it's less money consuming. It's also cheaper than in the sheep, and it is easier to apply the principles of the 3 R's: the replacement, the reduction, and the refinement, in order to have a better experimental animal conditions for the animals. It is very important to remark that the susceptibility to prion strain is governed by the PRP expressed in the animal, in our case, in the transgenic mice. For the generation of new PRP transgenic mouse model, mouse PRP was cloned in a plasmid vector, and this construct contains the genomic regions that control mouse PRP expression and can be excised from the vector with the NOT1 restriction enzyme. This is an endonuclease. This will allow us to purify the construct for the genetic modification of the mouse. And it is important to remark that with this construction, the open reading frame of the PRP is surrounded by the genomic region that controls mouse PRP expression. With molecular biology techniques, we can replace these open reading frames, in our case with a sheep, the open reading frame of sheep PRP, and we can generate transgenic mouse models with this construct that has several advantages. We, as mentioned before, the expressed PRP governs the susceptibility to prion strains. In this case, sheep PRP. This PRP is expressed under the control of the mouse prion promoter. So there is a similar space-temporal pattern of PRPC expression that in the wild-type mice. And that we can have a overexpression of the PRP, and this allows a certain incubation time, reducing the time that we need for this kind of experiments. These are adequate models for mutation studies in a controlled genetic background, because it always is the same, the genetic background of the mouse that we are using. As mentioned previously, we can excise the fragment construct by digestion using the NOT1 restriction enzyme.
Juan Carlos, excuse me, we are still with the first slide. Did you move forward?
Yes.
So let's try to close again, or I do in the presentation mode.
Maybe you have to change to a full screen or something on the PowerPoint.
Yeah, now you're. Could you change into the presentation mode, in the full screen mode?
Let's do it again. Let's see.
That's me. So, okay. Yeah, okay.
Nice, better.
Yeah, now you're going ahead. That's perfect. Thank you. Sorry, you are muted now. You're muted, Juan Carlos.
Can you hear me now?
Yes.
Okay.
But
let's try again. Sorry.
Yeah. No problem. I'm glad you are here.
Let's try again. We can digest the fragment with the open reading frame and the regions that controls the expression of the PRP gene with NOT1 restriction enzyme, and we can purify the band. In this way, we can a fragment of 12 kilobases that has not any bacterial sequence, and with this fragment, we make the microinjection of embryos derived from females that are cow for the mouse PRP, are PRP null mice. To establish new transgenic mouse lines, a few hundred embryos may be employed, wherein a select few will serve as founders once the construct has been randomly
I'm really
integrated.
sorry for interrupting, but we are still not moving in the slide. Should I take over with the presentation? So it seems to be a problem, and either you have the audio but not the slides, or the slides and not the audio. So I can give you the slides from my
Okay.
test, and so you have to tell me when I have to switch.
Okay.
Yeah.
I don't know why. Okay.
Do you see it now?
Yes.
Is that correct? Or are you another slide?
Yes. That's correct. Perfect.
Okay.
I don't know. So we have to establish a new transgenic mouse line. A few hundred embryos may be microinjected in order to select a few that will serve as founders once the construct has been randomly integrated into the mouse genome. Moreover, this plasmid may be modified via point mutation to generate new constructs incorporating the polymorphic variant of interest, which will be used in subsequent studies. In this way, the transgenic mouse lines generated can be considered identical except for the point mutation introduced in the PRP sequence. Next slide, please. Once the new mouse line has been successfully established through a rigorous process of genetic selection and controlled breeding, the breeding of homozygous transgenic animals becomes feasible, enable the maintenance of the mouse line under homozygous condition by one side, and also ensuring the production of a sufficient number of female animals for the subsequent inoculation of the offspring. It is important to emphasize that in our experiments, transgenic mice are intracranially inoculated with a specific isolate being tested. Although we know that this method does not reflect the natural route of prion transmission, it is considered the optimal approach for studying prion propagation. By introducing prions directly into the central nervous system, potential barriers associated with alternative routes are circumvented, also allowing precise control over the quantity and location of the prions administered to the mice. This inoculation route specifically targets the primary sites of the prion replication, the brain, where the highest levels of PRP expression are observed. So we consider that it is the best way to do the experimental inoculation of prions. Next slide, please. As you know, classical scrapie is a prion disease caused by various prion strains. In a study published 2 years ago, we employed 2 transgenic mouse lines that overexpress either sheep, wild-type PRP, or bovine PRP. This model served as a valuable system for discrimination between different classical categories of scrapie, so different classical scrapie strains. By examining the molecular mass of the PRP-RES and glycosylated band detected in the brains and analyzing the survival time in this model, if the disease occurs in a slow or a fast way, we were able to effectively differentiate at least 4 different classical scrapie strains. These specific isolates were carefully selected to represent the diverse range of classical scrapie strains currently circulating in continental Europe. Next slide, please.
Now I have the problems. Okay, just a second. Okay, now we don't know what's happening. Okay, I'm sorry.
Thank you. In the current project, our objective is to conduct a comprehensive analysis of the susceptibility to the 4 distinct categories of classical scrapie, so the 4 different strains currently circulating in continental Europe, using the newly generated transgenic mouse line. This analysis will also include the Icelandic classical scrapie isolates, which have been carefully chosen for the bioassay experiments. A comparative evaluation will be performed, comparing the results obtained in the transgenic mouse model that express wild-type PRP from sheep and this transgenic mouse model expressing the T137 sheep PRP. By conducting this study, we aim to gain valuable insights into the susceptibility patterns and potential variations among these classical scrapie strains. The epidemic BSE strain is also included in this study as a control. Next slide, please.
Today is the problem of technique and don't know what's happening. Okay.
In this prior study has been conducted to examine the role of the R171 polymorphic variant. This is the same than in the ARR sheeps, and the K176 polymorphic variant. That in a similar study similar to our current research focus. In this previous work, diverse range of susceptibility and resistance were observed across the different mouse lines that were analyzed. And the susceptibility was found to be strongly influenced by the specific prion strains that were inoculated. As expected, animals expressing wild-type PRP demonstrated susceptibility to all tested prion strains. And all tested genotypes of CPRP were also susceptible to a typical scrapie infection. However, mice expressing the ARR genotype, the R171, were unable to propagate efficiently the 4 classical scrapie strains that were assessed in this study. On the other hand, the resistance observed in relation to the K176 polymorphic variant depended on the specific classical scrapie prion strain that was inoculated. Notably, all the infected mice exhibited extended survival time when the K176 polymorphic variant was present, where the animals were infected with classical scrapie. Remarkably, the presence of the 176K polymorphic variant conferred resistance specifically to the epidemic BSE strain. These findings highlight the intricate relationship between different polymorphic variants and their impact on the susceptibility or resistance profiles across the different prion strains assessed. And finally, in the next slide, please. The open question to be answered is the role of the T137 polymorphic variant in the susceptibility or resistance to the different prion strains, with a focus, of course, on its role in the susceptibility or resistance to the classical strains circulating in Iceland. And we hope that this role will be clarified in this project. Thank you very much for your attention. Christine, you can go on if you want to, if you have any question, or we can talk on later.
I think we will deal with the questions later. Okay. I will go ahead with the talk as long as the technique is working. So. So you should see my slide now. Yeah. Okay. So in Work Package 2, we focus on the prion strain evolution, and in doing so, we would like to analyze the prion strain, the prion strain biodiversity in Iceland. And we know so far that the impact of selective pressure, for example, environmental factors or host genetic, is vital for prion strain evolution. And the special of the Icelandic situation is that we have 3 factors involved. We have time, environment, and host. So we can take isolates from different regions and years, from individual farms, from different years, from different sheep genotypes, and so on. And here you see an overview of our first selection, which these samples are already distributed within Europe, within the partners. And as you can see, we have all 3 factors involved. We have time, environment, and hosts in these samples. These samples will be examined first by biochemical examination by Romulo Nonno and Laura Pirisinu in Rome. They will undergo in vitro examination by PMCA on real-time quick in Scotland and France, and we will do from selected isolates a mouse model mostly with transgenic mouse models overexpressing sheep, bovine, human prion protein, and representing the species involved. And all these data will allow us to characterize the prion strain, and in particular how they evolve over time. And because we have the human PRP included as well, we will detect if there will be any zoonotic risk involved. The environmental factors are part of Work Package 3, and this was comprehensively presented in June and recorded. So I skip it to go ahead to Work Package 4, which is done by Jörn Gethmann. It's an epidemiological analysis of scrapie in Iceland, and he will develop economic models. And for this, he not only will obtain own data, but he will also use all the data which are coming from the first 3 work packages, and how he will proceed in that work package. It's up to him to explain tonight. So, Jörn, that is yours now. I hope the technique will work for you.
Thank you, Chris. So I will try my best. And I hope you can see the full screen mode.
No, yes, no. Not yet.
Okay.
Yes.
Now you should see the full screen. Yep. And Chris already told you that I will focus on work package 4, and we will use data from the other work packages in this. And I'm very happy that Angel did a very good introduction. And first, I want to tell you about the motivation. So very often in Europe, control programs are set up in a top-down approach, meaning that they are created by the administration, and farmers has to follow it. And in very often, some farmers don't agree and do not live the program. So after a couple of years, farmers might get frustrated. So as previously stated out, it's very important that it is in agreement between farmers and authorities. And what we have seen that it's always, and you can see it from the presentation of Angel, that the disease control programs need to be optimized after several years, and it is important to have a cost effectiveness analysis. So an analysis where you see the costs on the one hand and the effects on the eradication progress on the other hand, and not only in terms of costs, but in terms of what are the consequences, how many animals have to be killed, and what are the social problems of a program. And with our model, we want to show the consequences of different control programs. And a control program here means a strategy. And this is a combination of different control measures. And Angel already showed you 3 options, and they are combined with surveillance programs and with trade restrictions. And at the end, we want to see the time frame. How long does it take for different control programs to get to the end of the control? What are the financial consequences and what are the social consequences? And I can skip that because Angel and Chris explained it very well. And I want to show you what I intend to do from an example from Germany with a control of a cattle disease. It's bovine viral diarrhea in Germany. And we started with control of a BVD before 2000. And it was a control program in different federal states. And in 2011, we started with a compulsory national control program. And it was agreed between farmers' organizations and authorities. And between 2011 and 2016, the number of outbreaks decreased rapidly. But we still had several outbreaks per year, so single cases. And the disease control program, so each animal had to be tested after birth, was very expensive. So the aim of this study was what to do next. Do we need to change our strategy? What are the advantages and disadvantages of different strategies? And how to proceed. And it's just an overview. Here you can see the BVD control program. In number 3 was a control program that was done at the time of the study. And then we had different options to do to stop with a control program, to combine vaccination and the testing. I don't want to go into details, but I want to show you what was the effect of these control programs. In A, it was in 2011 when we started, and so you can see a rapid decrease. Only in strategy 1, where we did nothing, you see only a very slow decrease. And in 2016, we changed, we refined the control program. And here in 2017, we were thinking what to do next. And then you can see the different strategies, and some of them go to the bottom and we get free with them. Others will stay at a very low prevalence. And other control options are not doing very well. The number of cases increase after it and then go down. And so all these strategies had different consequences in the control program. And another point I want to show you is that this BVD control program, some of the most successful control programs here, it's scenario 8, were very expensive. They were successful, but quite expensive. And here you can see another point. So without doing anything, the highest costs are the direct costs, and that are the animals that are diseased or that die of the disease. So, and even if other control programs are not beneficial in terms of having a reduced cost to the compared to doing nothing, they reduce the number of infected and diseased animals. So it includes animal welfare and the animal health and the social aspect as well. So within work package 4, we want to we want to review the disease control programs in Iceland and Europe and the world. And we want to do surveillance, a questionnaire survey to get information from the farmers to get their point of view and to see the risk factors. In part 2, we want to analyze these questionnaire survey and find out the risk factors. And then we want to start this cost-benefit social analysis of future outbreaks and of the control program and give feedback to the scrapie eradication program in Iceland. And in detail, in part 1, we want to review the disease control programs, and what we want to do is we want to summarize the current disease situation, the situation on outbreak farms, and to get more knowledge about the risk factors. And to get this data, we want to perform a literature review on the one hand, and we want to exchange data with other work packages, for example, on the environmental risk factors. We want to carry out interviews with organizations, and we want to do farm visits with a questionnaire survey. And to do such a model, we need your knowledge to summarize the current situation and get more information about what has been done before and what is working. And for these farm visits and the interviews, we will use a questionnaire. We want to visit several farms. We want to analyze the situation on affected farms as well as on non-affected farms, and look at the farm management and on the control measures on these farms and the time course and outlook and perspectives for affected farms and even the perspectives on non-affected farms. And these farm visits will take some time for each farm because it is quite complex. And but we want to do it in an anonymized form, so all data we obtain will not be published. Only in a summarized form we will publish these data. So, and the second part, we want to use this data and data from other work packages to analyze the different risk factors and run a model to see which influence these risk factors have. And that's what we will do. We summarize the data from the farm visits, include data from work package 2 and work package 3, run a regression model, and eventually include results from previous studies. And all this data will be collected, and in part 3 we will find out what is the best strategy, a combination of control measures, what is working in your country. So we heard from Angel that the strategies are not working in each country in the same way. So what is the best for Iceland? And we want to do this cost-benefit analysis. And we want to define the different strategies, the control measures that will work for Iceland or probably work for Iceland in agreement with all the partners. And the outcome should be to show the time frames, how long does it take to get free, to show the costs of the different control programs and the social and animal welfare impact. Our time frame is that we create the questionnaire until February 24, and then we were thinking about running the questionnaire survey between March and April 2024, then to analyze it and give feedback to all the participants until the July 24, and then starting with the economic model in August 2024. And we want to be open and to discuss always the model with all the partners and to modify the model if something does not work. And we want to openly present all the results so that it is a discussion what would be the best strategy for Iceland, or probably best strategy. So that's from my side, and so I give back to Chris.
So thank you. Now I'm back again. So you should see that. Okay, so taken all together, using the data we obtain from the different work packages, for us in embryo research, we can test our hypothesis concerning embryo strain evolution, in particular how they evolve over time, the genetic and the environmental factors involved, all of that under field in the field, which is really special to us. And we will gain new insights into the role of the environmental PRPSC in the disease outbreak persistence. All we know based at the moment on experimental data, so that's very, very important. And we will gain knowledge of the multifactorial processes with which triggering the occurrence of zoonotic traits. And overall, it's scrapie in Iceland for prion research is kind of a scrapie in a nutshell. It's a blueprint for outbreaks of prion diseases occurring worldwide. So it's a lot we expect from our results. But on the other hand, there's a lot for you to gain, and these results have also the benefits bringing new breeding strategies, which could help to preserve your unique Icelandic sheep breeds. You will gain new insights into prion strain evolution, environmental distribution, which can be used for you to eradicate Icelandic classical scrapie more efficiently. And based on the models which Jörn will develop, the control programs can be further developed, can be adapted, can farmers and authorities can closely work together based on these models to get the best for Iceland. So I think that's a project where everybody has its benefits from, and we are looking forward to the next 3 years working closely with you. And I'm coming to an end. It's the circle is closed. That picture was taken on the information evening in Iceland at midsummer, midnight, and I thank all my colleagues involved in the Scrapie and Ice project, and I thank you for your attention. Juan Carlos, Jörn, and I'm open for questions. So just let us know if there's still something you would like to know? Sigurborg, yes.
Yes, thank you, Jörn. It was very nice to hear your presentation and to hear what you are planning to do. That will be very great, a great time when you finish your work. But we are planning to make a national scrapie program with new approach using the breeding. But if I understand you right, that you will be ready to tell in the end of year 2024 what will be the best model for Iceland. Is that right understood?
So yes, I understand that you already have your control programs and you started with a breeding program. So, but I still think that there are some questions that are working on a long term and might be helpful, even if the model is coming a little bit late.
Yeah. That's nice. So we will not just wait for the results from your work, but we will start, of course, but that will be very useful information that you will be bringing us in 1.5 years. So I thank you in advance.
Yeah, I hope that it will be very useful. Maybe one result is that the program you're doing at the moment or with your breeding program is the best option. But as we have heard from Angel, there are always some small side effects. Is it better to breed in start in one farm with a breeding program? How to do the trade restrictions? What to do with affected farms? Because if you keep all the animals on the farms, then they cannot trade anymore for a very long period. So all these details can be analyzed and tested. So for that reason, I think it will take some time to get with the breeding program to a success. I'm sorry. And so I hope that it will be helpful for you.
May I ask, Jörn, you will also take into account that codon 137 breeding strategies, it's that planned?
I think the first step is to get an overview on all these different options, and then to agree with the farmers organizations and the authorities on the strategies, what should be included in the different strategies and what is not realistic for Iceland. So for me, it's too early to say what we include in the model.
Okay. So any more questions? No, so everybody's tired. At least in our part. It's already 11 p.m. Okay. Then I would like to thank you everybody for coming, and again Angel for the very comprehensive information. I'm looking forward to seeing you hopefully soon, someday in Iceland again. Still remember the very nice business trip, the most fantastic business trip I ever had, and wish you all a good night. And yeah, thank you very much for your attention.
Thank you. Bye-bye.
It's a pleasure. Bye-bye.
Thank you very much.
Thank you so much.
Bye.
Thank you so much. Good
Bye.
night.
Good night.
Thank you.